This is Tom. Tom likes molecular biology and he has a DNA sample. Tom knows qPCR is a fast and sensitive tool he can use to quantify his DNA without having to do a post-PCR gel analysis. Tom loves how qPCR combines the exponential synthesis of thermocycling with simultaneous fluorescence detection, with the amount of product generated at every cycle being proportional to the amount of starting template, as well as the components found in a normal PCR mix. A qPCR mix includes a fluorescent dye to bind the double-stranded DNA. The dye is excited by a light source, and as DNA is synthesized, a detector records the increasing signal. A qPCR experiment consists of 3 phases. However, the reagents are abundant within the exponential phase, providing the best window for accurate quantification. The point at which fluorescence can be measured without background noise affecting accuracy is called the threshold line, and the number of PCR cycles at which Tom's sample crosses the threshold line is known as the CT value. 2 major types of qPCR can be used to assess a sample: absolute quantification and relative quantification. Using a sample with a known number of template, Tom can perform serial dilutions and perform qPCR using these known standards to obtain CT values for each dilution. When these CT values are plotted against the log of the initial copy number, a standard curve is created that can be used in absolute quantification to determine the starting copy number of an unknown sample. An advantage of this method is that it does not require validation and optimization using a housekeeping gene. However, reliance on serial dilutions in this method may introduce error. Separate curves are also needed for both the target and control. Using this method, Tom could also analyze the rate of gene expression by first reverse transcribing an RNA sample into cDNA, amplifying it with qPCR and plotting the fluorescence on a standard curve as before. Relative quantification can be used to quantify the difference in expression levels of 2 or more transcripts. The initial copy number is inversely proportional to the number of cycles required for application to exceed the detection threshold. A low abundance transcript will require more cycles, whilst a higher abundance will require fewer cycles. The use of more than one validated reference gene will generally result in more accurate results. Both the target and the control can be amplified in the same tube in relative quantification, eliminating the need for a standard curve serial dilutions required for absolute quantification. However, this method has the added complexity of requiring housekeeping genes as standards to account for variability between samples. Housekeeping genes are considered as constitutive genes, where they are transcribed continually as opposed to facultative genes. They are also expressed by all cell types and do not vary under experimental conditions. Tom knows that some commonly used housekeeping genes are the TATA box binding protein, ubiquitin C, and the MHC class I. From the CT values we obtain from the qPCR, we can then compare the 2 genes using 2 to the power negative delta delta CT, where we subtract our delta CT for our control sample from the delta CT of our experimental sample. We then compare the regulation between our 2 experimental conditions. This gives us the amount of up or down regulation of our gene of interest.